Welcome back, folks. This is Chapter 4, Gene Interaction. The chapter is essentially broken up into 4 parts. Part 1 looks at the interaction between alleles and how they produce a dominance relationship, looking at their biochemical makeup as well. Part 2 looks at genes that produce variable phenotypes, so not always dominant, but something different. Part 3 looks at gene interactions that modify Mendelian ratios. And the last part looks at how to distinguish if mutations are in the same gene or different genes, something known as complementation. All the slides, 94 of them, are fair game for examination. In most cases, students have been looking at examples of genes that have 2 alleles, for instance, big B, small b. There was one case earlier on that we looked at, the blood types ABO, and we'll bring them back again. But ABO is a representation of what we need to learn. There are many genes in the population where there are more than 2 alleles. Understanding the consequences of having a particular combination of alleles is one of the requirements of learning this chapter. Second, dominance of one allele over another may not be complete. So partial dominance, codominance, incomplete dominance. So we'll have to learn all these terminologies and, more importantly, how they work at the biochemical level. For many traits inside the human body, we are discovering that more than one gene is responsible. So 2 or more genes may affect that single characteristic. Understanding that system is also necessary. The last bullet point simply takes the third bullet point and expands upon that. What it's saying is that the expression of a trait may not only depend on more than one gene, but it may also depend on the interaction of the genes and their products with non-genetic factors. And this is what we call the environment. The documented component of Mendel's work shows that he chose to work on traits that happen to have just alternative, easy to identify forms. For instance, yellow seeds, green seeds, tall plants, short plants. So in each of his cases, documented cases, one form was completely dominant over the other. That meant that in the heterozygous state, big A, small a, the phenotype of the dominant could not be distinguished from big A, big A. It appears that in the human condition, about 1/3 of our genes are responsible for controlling directly 1/3 of our characteristics. But the majority of our genes work in more complicated ways. Of those 1/3, how many have just 2 alternative forms? Very few. Most of the genes that we have that are Mendelian in nature have multiple alleles. We now have to correlate the phenotype of an organism, what's observed, with what's happening at the molecular level when it comes to expression of genes through transcription and then maybe translation, and how those products interact within cells to eventually lead to the phenotype that we're studying. In most cases, it's the protein product of the gene that somehow interacts with the biochemistry of the cell to realize the phenotype. So the overall phenotype is a consequence of the activities of many different proteins in most cases, and realizing that the alleles of a particular gene will make different varieties of these proteins. Example 1 tries to apply some mathematics to this biochemical conundrum. We are looking at one gene with 2 alleles. The wild type allele, the one that's most common in the population, that's called the wild type, is designated big R+. And the alternative allele, the mutant allele, is designated small r. Assume that the wild type allele produces 50 units of enzyme per cell. The mutant allele is so defective that it doesn't produce any functional protein, therefore 0 units. Also assume that 40 or more units of enzyme activity has to be present in order for the wild type condition, the phenotype, to manifest. Therefore, when we are big R+, we have a total of 100 units, well in excess of the 40 units required for that phenotype. And believe it or not, with the big R, small r combination, we still produce 50 units from the big R+ allele, enough to satisfy the needs. But the problem arises when we have two small r's. We're in the homozygous recessive condition. We don't produce any units of that enzyme, which results in the mutant phenotype. So this is one way to relate the presence or absence of enzyme to the terms recessive and dominant. Incidentally, there's a genetic term for this kind of relationship with the heterozygous. It's just as powerful as the homozygous on the phenotype. And we call that a haplosufficient allele. So the dominant wild-type allele is haplosufficient in that just one copy of it is enough to encode normal function, even in the case of the heterozygote. Example 2 allows us to progress into another arena of genetics that we'll talk about as being haploinsufficient. That's coming up on the next slide. Let's put the scenario in place. This time, let's look at another gene with a different designation. We'll use the letter T instead of R in the previous example. So assume once again that this gene has two alleles, T1 and T2. Now, this is an alternative way in which genetic alleles can be represented. The use of the subscripts is appropriate. So T1 is one allele and T2 is the other. Let's look at the relationship between these two alleles. Assume that T1 produces an active enzyme with 10 units of activity, and a mutant allele, T2, produces less enzyme activity, this time 5 units. And assume that 18 or more units of enzyme activity will produce the wild-type phenotype. So we can see in the homozygous dominant condition, we have 10 units from each allele. So overall inside the cell, we produce 20 units of enzyme, more than enough to manufacture the wild-type phenotype. The problem lies with T1, T2, which is the heterozygous condition. That only produces 15 units. And of course, the homozygous recessive T2, T2 will only produce 10 units because it's 5 units per T2. So individuals with this will have a mutant phenotype because neither possesses or produces enough enzyme to exceed the threshold of 18. From the mathematical calculation and from the biochemical calculation, there's not enough product produced by this biochemical pathway to make the phenotype wild type. We see that the heterozygous condition is very much the same as the homozygous recessive mutant condition. So what do we do in this case? Well, we have to be observing what's happening. So the dominant allele is the mutant allele, because in the heterozygous condition, it produces a mutant phenotype. So this now helps us understand dominance versus recessiveness. And in some cases, the heterozygous is dominant, and in other genes and their alleles, the heterozygous may be recessive. With example 2, one copy of the dominant allele is not enough to give you the phenotype, so we call that haplo, one copy, insufficient. You need both copies to be dominant. An overarching conclusion of the way that alleles interact for different genes has led to some patterns. Non-wild type alleles can produce biochemical effects in various ways. One of them is called a loss of function effect, in which the allele causes a decrease or complete loss of the function of that gene's product. And the alternative class is gain of function, where the presence of that allele now acquires for the cell a new function or increases the activity of an existing function. Figure 4.1a is a graphical representation of a wild-type scenario. So here we have the product inside the biochemistry of the cell, and here we have the genes inside the nucleus of that eukaryotic cell. So if both alleles are normally functional, each allele will produce some units of the product. So in this case 2 units, and in the case of the second chromosome 2 units as well. And these are enough to allow the cell to have a normal phenotype. Those mutations that cause a complete loss of function, i.e. no functional gene product, have a name. They are called null mutations. Null means none, so nothing is produced by the allele. But there's a genetic term too, and it's called amorphic. So these are amorphic mutations. Often they are lethal when they're in the homozygous condition. The alternative situation, which results in a partial loss of function, we call those leaky mutations. But in the genetic terminology, they're called hypomorphic mutations. Hypomorphic. So not enough. And the severity of the phenotype depends on how much of the activity still exists for that cell. Figure 4.1b shows both the amorphic and the hypomorphic condition. So the X's represent mutations. So in the homozygous condition, there's a complete loss of function, and there is no protein product. In the heterozygous condition, one allele, the top one, is mutated, the bottom one is wild type, and the wild type produces some product. And in the second example below, we have a loss of function of the hypomorphic type. So the light color represents some function of the enzyme. So the two alleles here in the homozygous condition produce something, whereas in the heterozygous condition, they produce just more than this side, but not enough to make the situation completely wild type. It is important that you grasp the next concept, and that is of dominant negative mutations. A mouthful, but quite easy to understand. This applies to proteins that are a result of many polypeptides working together. So a protein is the finished functional entity, but that protein may have components, and the components may come from different genes. So we call those multimeric proteins. The reason these mutations are negative is because they have a detrimental effect on the phenotype of that cell. And the reason they're called dominant is because all it takes is one gene product to be defective, and it can spoil the good stuff that the other gene is producing. Thus, the combination of the two is non-functional. It's like having two genes make two parts of your car, the front of your car and the back of your car. If the front of your car is okay and the back of your car is mutant, then your car is not going to get anywhere. Let's look at figure 4.1d. It can be a little confusing, so let's start by dissecting it. Let's begin at the end, like right here. So imagine that this cell needs this protein, and this protein is a combination of the brown and the purple. Each one is produced by its own gene, but they come together, the polypeptides, to form the final protein that works nicely. So assume that the purple product comes from its own gene that's not shown, and that gene is not part of this analysis, and that gene is going to be perfectly normal. So it always produces a normal purple. But the other half, the partner, this polypeptide in brown, it comes from this gene, and this gene is in the heterozygous condition. So the bottom allele, the wild type allele, produces a wild type polypeptide, the brown one, and that then combines with the normal purple to produce perfectly normal proteins. That's what normally should happen inside the cell. In the heterozygous condition, the top allele is a spoiler. It produces a polypeptide that has a mutation, indicated by that black mark. The cell will now produce both types of polypeptide from this gene. The bottom one produces normal, the top one produces mutant. Because either one of these can combine with the normal purple polypeptide, in some cases we end up with this, if the bottom polypeptide combines with the normal polypeptide there, and in other cases we get this, an abnormal non-functional protein, because the top brown allele causes the purple allele product, that polypeptide, to become malformed, and that protein doesn't work. So in a heterozygous condition, we have both types of product, the normal enzyme and the defective enzyme. And this, of course, will interfere with the chemical reactions, because sometimes this enzyme gets to the site of the chemical reaction, and other times this one does. And that will reduce the overall efficiency of the cell. And this would act as though this was a dominant mutation. The left-hand side of the slide simply shows you what happens when the second gene is in the homozygous condition and its polypeptides are 100% mutated. Therefore, every time they interact with the normal polypeptide in purple, they will always produce a non-functional or abnormally functional protein enzyme. Drilling down into the gain of function mutations, they also occur in two general formats. They may be hypermorphic, where they produce more gene activity than the wild type, or they may be neomorphic. Neo means new. They may have a novel function that the cell has now acquired because of this mutation. In most cases, these types of mutations are usually dominant over the wild type. Figure 4.1e tries to convey that. Let's look at a hypermorphic mutation. The wild type would be two normal genes, which is not indicated here, but that would produce 4 units of enzyme. Then if we have a mutation in one gene allele, then we have 5 units of enzyme per cell. That may give an advantage. And if we have both alleles mutated, we may get 6 units of product. Now sometimes this may be too much for the cell to handle, and this condition can be lethal. But we'll look at that in the future. Let's move on to the bottom part. This is again a gain of function, but this time there's a new function gained by the cell that didn't exist before, and that's indicated by a bend in the product. So there would be 2 normal alleles here, and they would produce 2 sets of straight product. Now the mutation creates an abnormal function that gives the cell an added benefit. And in this case, in the homozygous state, both gene products have given the cell a new function. Now the severity of this depends on what the original function was. If the original function has been overtaken by a new function, the cell may survive. If the original function has been replaced by a new function, the cell may not survive. There is one area of genetics that's causing concern for everybody. That is the use of notation when referring to alleles and wild type and mutants. Because many different scientists used alternative, random ways of expressing what a mutant allele was, there's no standardization. Different textbooks today will use their own combination of rules, so please be on the watch out. The rules in this textbook were chosen by the authors, and they represent their version of how to notate genes. And that means it's dependent on the species that the original scientist identified. So those legacies have come through in a species-specific manner. And you'll notice this throughout this book. Other than the very first example that we gave you, all the other examples produce alleles that produce some variation in product, other than the null mutations. So this type of interaction between alleles, where the heterozygous is somewhere between the homozygous on each side, is called incomplete dominance. So incomplete dominance has another name called partial dominance, but incomplete dominance is what we tend to use generally in genetics. The heterozygote may not be exactly in between the two extremes of the two types of homozygous. It could be closer to one than the other. So here is flowering time in pea plants, very similar variety to what Mendel used. And you can see the homozygous 1 condition is over here, and that flowers on the first day. And the alternative homozygous flowers on the 5th day. Whereas the intermediate, the heterozygous, we would expect to flower halfway between the two. But in this case, it tends to flower almost close to 4 days. So a great example. The next category is codominance. This occurs for some genes. And an example is the ABO blood system. And this is a bit more complicated than simple codominance. But let's define codominance and we'll come back to the ABO blood system. So in this case, if you're heterozygote, you will express both the dominant and the recessive alleles at the same time. For instance, if one chromosome carries the A type of marker and the other chromosome carries the B type of marker, you will not just produce A or B, you'll produce both in equal numbers. Both A and B markers will be found on the surface of that cell. Thus, the phenotype of the heterozygote is different from either homozygote. The human ABO blood group system has 3 alleles. They are designated IA, IB, and lowercase I. The IA and the IB are equally dominant over each other, so they're co-dominant, whereas each is completely dominant over the small I allele. So we have a mixed. An understanding of the biochemistry behind this makes everything perfectly clear. So let's start looking at that. So blood type A involves the presence of an antigen on the surface of red blood cells. Blood type B produces an alternative shaped antigen that's present on the surface of red blood cells. But blood type O doesn't produce anything that's detectable on the surface of the red blood cells. So blood type AB people have both antigens, and blood type O people have neither. The slide here shows you the relationship between the type and the possible genotypes of that individual. So this is the phenotype, and these are the alternative genotypes that will give rise to that phenotype. This should make sense to you based on your previous genetic understanding. The next slide simply shows us what happens when blood of one type is mixed with blood of a different type. Antibodies are produced by a body against a particular type of antigen. So the anti-A antibodies will bind to red blood cells that carry the A type of antigen and cause those cells to clump. And antigen B will do the same thing if the surface of the cell, the red blood cells, is carrying antigen type B. So let's see what happens. So if blood is taken from a patient with type A and then subjected to 2 test tubes, one containing anti-A and the other anti-B, then clumping will only take place in the first test tube where the antibodies are looking for A to clump together. In B, there is no clumping because these antibodies cannot find a B antigen on the surface of those red blood cells. In the same vein, let's go to the last example. We have anti-A antibodies, and they're looking on the surface of blood type O for A, and they can't find any because type O blood has neither A or B. Then when you put anti-B into a test tube and combine it with type O blood from a patient, there's no clumping either. So that tells you the phenotype, and from the phenotype you can deduce the genotype. So the interesting one is AB. So patients AB, they will produce on their surface both A and B antigens. So anti-A will cause clumping because it binds to the A and recognizes that, and anti-B will cause clumping because it recognizes the B and binds to that. What you may not know is the biochemical nature of the antigens. So the antigens are called glycolipids, and these glycolipids are on the surface of red blood cells. So the lipid part is embedded in the phospholipid bilayer, and the sugar part is added to the exposed surface of those lipids. Therefore, when somebody talks about blood ABO types, they're looking at not proteins, but glycolipids. Want to know more? Let's go one level deeper. The sugar component of the glycolipid is illustrated here. So this is the basic structure of the sugar that's applied to the lipid component of the glycolipid. Now these sugars can be modified by 2 enzymes. The first enzyme, called the A transferase, will transfer here this green molecule. So this will make the surface of this sugar the A antigen. If a patient possesses an alternative form of the enzyme, the B transferase, and that's encoded by the IB gene, then their cells are able to transfer a sugar group here, again onto the terminal part of this molecule, and you can see that in blue. And if a patient has two broken alleles, they will not do anything to this molecule. So now you can see the relationship at the chemical level, the biochemical level, and at the phenotypic level of the ABO blood groups. Let's move on to the next concept, that of allelic series. An allelic series is simply the order in which the alleles affect the phenotype and which one is dominant or codominant over others. Let's begin by looking at a term. A very common finding in genetics is that a gene may have more than two alleles in the population. So those are called multiple alleles of the same gene. If a gene has multiple alleles, then the question becomes which one is dominant to which. And that's nicely illustrated in this example for mammalian coat color. So the C gene system is what we're studying. Many mammals with coats, such as cats, mice, they have a C gene. C gene stands for color. And it produces an enzyme named tyrosinase, which is active in the production of melanin. Melanin is the dark pigment in skin. And the gene has dozens and dozens of alleles, many alleles in the population. But four of them form an allelic series, as discussed on the next slide. So the wild type allele is called C, big C. The next allele is called small c, ch, for chinchilla. And that produces, as we'll see here, a dilute coat color. The next allele is called small ch for Himalayan, and that produces this very, very characteristic coat color. And the last allele that we'll consider today is just called small c, and that's a null allele. A null allele is a dead allele, and it doesn't produce any color whatsoever, so the creature is an albino. Even the eyes are pink because there's no pigmentation in the iris. So these four alleles have a relationship that's been worked out, and that's called the allelic series. So here the big C is completely dominant over the others, and then the question is, what's the relationship between the others? And it seems that from this chart that chinchilla is dominant over Himalayan and albino, with Himalayan being only dominant over albino. The actual relationship between the alleles is a little bit more complex than we just saw. So here's a breakdown. So the big C allele is completely dominant over all the others. The chinchilla allele is partially dominant over the Himalayan. And all the other alleles are completely dominant over the small c. In order to understand this, we are given 6 crosses, A to C on this slide, D to F on this slide. And what we are required to do is to relate the color in the offspring to the color of the parents, therefore deducing the relationship between the alleles of this gene. We should have realized through pattern observation by now that if you have 3 generations of creatures that are diploid and you breed them together in a particular order, starting with pure breeding, and then you have a hybrid, and then you cross the hybrid with itself, you always get patterns. And those patterns indicate the relationship between the 2 alleles that you're studying. So a 3-to-1 ratio indicates dominance. Anything that departs from the 3-to-1 ratio will indicate some kind of alternative relationship between those alleles. So for crosses A, B, C, D, and E, we're getting a 3-to-1 ratio. So they indicate a standard Mendelian association. But cross F is a strange one. It gives us a 1-to-2-to-1 ratio between the offspring. And that is something to do with partial dominance. Notice that this box is chinchilla with Himalayan features, as is this one here. So here we have an example of both traits showing themselves in half the children, and therefore we can say that is partially dominant. Chinchilla is partially dominant to Himalayan. Once again, understanding the biochemistry makes things more obvious than just looking at the phenotype. So let's equate the two together. The C allele, the big C allele, produces an enzyme called tyrosinase that is 100% active in these cells, whereas the chinchilla allele is much less active. Thus, having one large C inside a cell is enough to produce the full coat color. When it comes to chinchilla, it depends on its genetic association with other alleles inside that cell. An interesting aspect of the Himalayan allele is that it produces an enzyme that's temperature sensitive. So in those parts of the body that function at higher temperatures, it is malfunctioning, non-functional. But in parts of the body that are at lower temperatures, such as the extremities, the tail, the ears, and the paws, then the enzyme is functional there, then it produces a black phenotype. The C allele is a null mutation and therefore produces no functional enzyme, therefore doesn't contribute to any coat color. Since alleles affect the biochemistry of a cell, there can be some instances where the product or the lack of product is dangerous and it causes the death of the cells and maybe the organism. And we do see those type of alleles present in the populations. These are called lethal alleles, and they are normally recessive and inherited in a way that the homozygote condition dies. So 2 small a's will be lethal. These tend to be found at 2 levels. The first level is the embryo, so once fertilization has ensued, and then during the development of the embryo, the lethality manifests itself during some phase of development, and that's when you get the lethal effect. In other cases, the gametes are lethal. So the gametes fail to generate properly if they carry that particular allele. So we call those gametophytic lethals. The embryonic lethal alleles can be detected by the presence of a 3:1 ratio in the living seeds versus the dead seeds. And amongst the living seeds, only these ones, the ratio 2:1, in that 2 are heterozygous and the 1 is homozygous. Figure 4.7 supports the lethality of alleles in plants. Here's the wild type condition, where the embryos are fully functional. Here's the lethal gene kicking in and killing these seeds. And here we can see the gametophytic lethal and the ratio that it produces. To reiterate, the ratio that we expect to see here is a 3:1. So for every 3 seeds alive, there should be 1 that's dead. And the ratio here that we expect to see from this cross is 1:1. You can see the 4 living seeds, and you can see the 5 or so dead seeds. The classic examples used in animals are to do with agouti. Agouti is the coat color of mice. It is a biochemical product of 2 pigments released into a single hair shaft, as indicated in the top half of this picture. The alternative allele is called yellow, and that just produces hair shafts with a yellow pigment, but no black pigment. In this system, the AY allele, the Y for yellow, is dominant over the agouti with respect to coat color. However, it is lethal as the homozygous recessive condition. So let's show you that with this diagram here. So we're breeding a pure breeding agouti mouse with a yellow mouse. And these are the gametes produced by each of those parents. And these are the possible children. And the ratio is 1:1. When 2 heterozygous yellow mice are bred together, they produce the following Punnett square. The surviving mice belong to these 3 boxes. And the ratio between the survivors is 1 agouti to 2 yellows. So no longer do you get 4 boxes, because the 4th box is a lethal. These mice are never born, and therefore never seen as pups. So when something like this happens in genetics, when something is not part of the equation, we can remove that. Therefore, we are just left with 3 boxes, and the ratio that we produce will be out of 3 and not 4. Upon further analysis, the AY allele is really a mutation, and that mutation is a deletion that affects a part of the chromosome that has 2 genes residing on it, agouti and Raleigh. And Raleigh produces a protein essential for embryonic development in mice. and the deletion connects the rally promoter to the agouti gene. So the two neighboring genes are rearranged such that the promoter for rally is now on the agouti gene. And in the heterozygous condition, an excess of yellow pigment is produced. Whereas in the homozygotes, they die due to the lack of the rally protein. So one rally protein from one normal chromosome is enough to allow the development of the embryo. But if both chromosomes have the deletion, then the rally protein is no longer made and embryonic development cannot pursue. Figure 4.10 provides a graphical representation of the chromosomes in their two states. So this is the A allele, where you can see the rally gene is in green and the agouti gene is in purple. And they both have their promoters. The AY allele has a deletion, which places the rally promoter upstream of the agouti gene. Therefore, the rally promoter is controlling the agouti gene, and the rally genes itself, the coding region, is gone. Another observation with genetics is the age of onset of a particular disease. So some dominant lethal alleles can sidestep natural selection. That means they can not be selected against because the children are already born and the genes and their alleles have been passed on to the offspring. And a classic case of that in humans is Huntington disease. We have learned earlier that it's a fatal neurodegenerative disease that develops at an early age and continues to affect the population into old age. And what happens here is that the symptoms of Huntington disease are mostly manifest in people's 40s and 50s. And by the time they reach the age of onset, they've already had their offspring, and the genes have already been passed into the next generation. Another genetic observation is that of variable phenotypes. So these individuals may have the same genotype, but the outward appearance is variable. Their phenotype is variable. The baseline is the same. For a given genotype, we assume that it always produces the same phenotype. And if that's the case, then we call that complete penetrance, because the genetics has penetrated completely into the phenotype without any side tracking or issues. In conditions where that doesn't happen, of which many exist, as we'll see shortly, then we can use an alternative term. So we find incomplete penetrance in sex-linked traits, sex-influenced traits, incomplete penetrance, and variable expressivity. Let's take a look at each one of these. Let's start with sex-limited traits. The genes are present in both sexes, but because of the hormonal difference in one sex versus the other, that gene is expressed in one sex and not the other. The book gives us several examples so we can relate to this. The first example is to do with breasts and milk production. So only females have the appropriate hormones to produce breasts, and they also have the appropriate hormones at the right time to produce milk. But males can't, even though both sexes carry the genes for milk production. The second example is horn development. That's limited to males in some sheep, cows, and other hoofed animals, even though the genes are present in both sexes. And beyond that, there's some behavioral traits, especially when it comes to mating behavior, that are strongly influenced by the hormones of the creature and their sex. So sex hormones differentially influence the expression of genes because they interact with the biochemistry within those cells and allow those genes and their products to be expressed. We should now understand what chemical castration means. You remove the hormones and then the behavioral desires can be removed accordingly. With sex-influenced traits, a particular genotype will produce different phenotypes depending on the sex of the animal. This is a great example given here with the chin beard. So in both sexes, you can have the homozygous 1 condition or the homozygous 2 condition. In the homozygous 1 condition, the animals, whether they're male or female, will be beardless. In the homozygous 2 condition, the individuals will be bearded, and that's both males and females. But in the case of the heterozygous condition, that depends. So if the heterozygous condition is a male, he will have a beard. If she is a female, then she will not be bearded. She'll be beardless. Therefore, the heterozygous condition is different among the two sexes. Figure 4.12 shows us the genetics of a cross between a beardless female and a bearded male. Thus, depending on which sex chromosome also accompanies this particular gene, the result will be a differentiation in the sexes in these two boxes. Let's look next at incomplete penetrance. As we mentioned earlier, an organism that has a direct correlation between a genotype and a phenotype every time with that variance is called a penetrant trait. However, if an organism does not always produce the exact same phenotype, generally from the associated phenotype, then in genetics we call that non-penetrant. Non-penetrant. In a population, you can have a particular genotype, and that always expresses the same phenotype, and therefore you can use the term fully penetrant for that individual. But a cousin may have the same genotype but produce a slightly different phenotype, and in that case we say that that individual has incomplete penetrance. The best way to study this is to look at an example. So polydactyly is the example of humans where they have extra fingers or toes. It's an autosomal dominant condition, so a big letter will make you susceptible to this. The most common manifestation is an extra digit on one of the fingers, and therefore leading to 6, as indicated in figure 413. However, when one looks at a pedigree of a family that has polydactyly, we find that they are individuals, such as number 6, 26, and 210, who should have had the dominant allele and therefore manifest polydactyly, but they didn't, and that's relayed by the shape of the family. So 3, 10, 2, 10, 2, 6, they should have had this, but they don't. So in that case, the dominant allele is non-penetrant. And that happens to be common in about 1/3 of the people that carry this. The next condition to relate to is variable expressivity. In this particular case, individuals that carry the alleles for the trait show a phenotype, but to a varying degree compared to other members of the same family. A classic example used in genetics is Waardenburg syndrome, which has 4 principal features. Each family member with this syndrome has the same genotype, but they may show a different combination of symptoms. The key at the bottom of this figure shows us the 4 principal features of this syndrome. So premature graying, white forelock, hearing loss, different colored eyes. So those segments of the symbols will indicate that particular combination. So the grandmother here, she has all 4 features. Her daughter only has 3 of the 4. And her granddaughter and grandson, they have different half combinations. You can see how that penetrates through the family. Different features for the same syndrome, which we say is variably expressed. One overarching principle of genetics is that from gene to phenotype, there's also a large component that we call non-genetic, and that is the environment. So gene-environment interactions are huge and they occur at many different levels. Stated another way, we can come to this. So genes alone are not responsible for all the variation seen among organisms. So this is the influence of the environment on the expression of genes at the biochemical level. The understanding of this interaction has been profound in preventing some human disease at an early stage. So the classic example is phenylketonuria, PKU. So it's an autosomal recessive condition, and it causes the absence of an enzyme that is involved in breaking down an amino acid called phenylalanine. So when infants are first born, they may have a lack of this enzyme. They may have PKU, and that is quickly detected by a heel prick and a quick test. If the patient is not detected in time, the buildup of phenylalanine inside the body becomes toxic, especially to neurons, and that can cause developmental issues. The prevention of this type of neurological failure is straightforward as far as the doctors are concerned. They just prescribe to the patient a diet that restricts the phenylalanine for the rest of their lives. The control over the levels of phenylalanine is all that it takes not to develop the phenotype. The next concept is pleiotropy. So pleiotropic genes have an influence not on one characteristic, but multiple characteristics of the organism. Thus one to many when it comes to genotype to phenotype. The most common occurrence of this type of gene is during development. And there's a classic example in Drosophila where the hormone called juvenile is having numerous effects on development. So if that juvenile hormone is defective, then you'll have impacts on body size, development time, sexual maturity, and on and on and on. A single gene with multiple effects. In the case of humans, sickle cell disease comes under the same title. So sickle cell disease is an autosomal recessive condition where a protein called beta globin is mutated. And beta globin is an important component of the hemoglobin molecule in terms of carrying oxygen around the body. So many red blood cells or people with sickle cell disease take on a sickle shape and cause numerous physical problems and complications. So a single mutation can lead to a devastating set of outcomes, not all of which may be present in one individual. The third section of the chapter goes back to looking at Mendelian genetics, but now it's looking for deviations from the ratios that Mendel obtained. For most of our characteristics, a single gene is not responsible, but a symphony of genes and their products is what allows us to produce those characteristics. So gene interaction is the norm rather than the exception. If one recalls their biochemistry course, they may remember that there are many branches and many steps to most biochemical reactions. In fact, the true picture is even more complicated than that. Many branches, many pathways, they impinge on each other, and they have ratio-dependent outcomes, negative and positive feedback. So we are very, very complex within our cells. So no wonder that different gene products can interact. But at the heart of everything, we have two types of biochemical processes. Those that build molecules, anabolic pathways, and those that dismantle molecules, catabolic pathways. And the interaction between them is very much known to a large degree, although there are some aspects that are still missing. In addition, we have signal transduction pathways that carry information into the cell, and we have developmental pathways which influence the relationship amongst different types of cells and their development. Figure 417 illustrates in red blood cells how sickling takes place over a range of phenotypic consequences. So you can see we have genes, and the genes produce products. In most cases, they are enzymes, and those enzymes then convert one substrate to a product, and the product becomes a substrate for the next enzyme. In many cases, we have branch structures, and you can see how different phenotypes here can manifest when it comes to broken genes or enzymes. The understanding of these pathways was first brought to the community by the work of Beadle and Tatum, who biochemically analyzed the fungus Neurospora crassa. It was they that first proposed the one gene, one enzyme hypothesis based on the evidence extracted from their experiments. What they basically said is that each gene produces an enzyme. That enzyme having a specific role in the biochemical pathway that they studied. Every time they produced a mutant phenotype, they could attribute that to the loss or malfunction of a particular enzyme in that pathway. So each enzyme defect was inherited as a single gene defect. So a prototype is an organism that has all its enzymes working, so it's wild type. An auxotroph is a feeding mutant. It can't feed unless it's given the missing substance that the enzyme doesn't convert. Keep in mind though that not all gene products are enzymes. Although the majority are, others include transport proteins, structural proteins, and regulatory proteins that control processes. But beyond that, believe it or not, most genes may only be producing RNAs. And that's what seems to be happening. As we discover the functions of more and more genes, it's clear that many are producing various types of RNA, not messenger RNA. In addition, as we saw earlier, that some polypeptides, the products of genes, may need to combine with the products of other genes to produce the functional protein or functional substance. Beadle and Tatum used an approach known as genetic dissection. In this, an experimental process is designed to investigate the steps of a biochemical pathway. It can be biosynthesis or biobreakdown. The work of Horowitz demonstrates this. He was also working on Neurospora crassia, the fungus, and he was aiming to discover the pathway responsible for the generation of methionine, a amino acid. And he wanted to determine the steps leading to the production of methionine inside this fungus. His goal was to identify every step by using mutations. The use of an analogy is useful in this particular scenario. So for some students, the analogy may relate. So let's look at a reservoir on top of a mountain, and it feeds a river. And along that river, we have dams, and the dams are either open or closed. So in order to get flow into the ocean, the dams all the way down the river have to be open. And that river represents a biochemical pathway. And each dam represents an intermediate step in that biochemical process. And the opening and closing of the dams is the functionality of enzymes in these biochemical pathways. So if an enzyme is present, it will open the dam. If an enzyme is defective or missing, it will close the dam. So let's start with the analogy. Let's say the first dam at the top of the mountain at the reservoir. That dam has an enzyme, and that enzyme is defective. So that means the dam is closed. Therefore, everything downstream from that dam is now dry. There's no water flowing into the ocean. So this analogy suggests that the final product, water in the ocean, doesn't happen. Therefore, the organism may suffer. However, as an alternative to overcome the dam at the beginning, we could feed the river with a stream just below the dam. So we can pump water from the stream into the river, and there will be flow into the ocean, therefore overcoming the closed dam at the top of the mountain. So that same analogy can be applied all the way down. Say we have 10 dams down to the ocean, and the fifth dam is now blocked. We have no enzyme, so the fifth dam is now closed. So if we put water upstream in dam number 2, that water will flow through dam 2, through dam 3, and then when it hits dam 4, it will then be prevented from flowing downhill. So dam 3 will pile up water behind it. So there's no water going into the ocean. So that organism will therefore have a similar outcome to the first that had no water going into the ocean. However, if we put water in after the dam, then there will be a flow into the ocean, and the phenotype of that organism should be normal. So there's the analogy. In the case of Horowitz's experiments, he was using food. Food was analogous to water. And the food or the biochemicals that he used were designed to overcome these blocks. So mutants blocked at a step after the intermediate, after the feeding of the river, still could not grow. Mutants blocked at a step before the intermediate, i.e. a dam before the stream feeds the river, will still have water flowing to the ocean. So by doing various experiments, he was able to deduce the order in which certain enzymes and mutations correlated. So let's have a look at the data that he generated. The important thing is the prototroph. Prototroph means an organism that has no mutations for this biochemical pathway. So it doesn't matter whether you grow it on the starting material from the top of the reservoir, or you give it food, feed it streams from various points along the river. It doesn't matter. It will always have water flowing into the ocean. So that's what a prototroph is. It can overcome any hurdle. Therefore, the mutants can now be tested one at a time. So let's look at methionine 1. So it can't grow on minimum medium. That means it can't grow by just being fed water from the top river. There's a dam that's blocked somewhere and water is not getting to the ocean. So then you supply it with a stream that overcomes the first dam, and it still can't grow. Then you give it a stream that overcomes the second dam. It still can't grow. And eventually you find that if you feed it a stream close to the ocean, that's below the dam that's defective. Therefore, it will start growing. So now you've discovered which dam has been blocked in that mutant. Do the same analysis for the other 3, rearrange them, and you find out that Met4 mutants, their dam is very early on. That's blocked in their pathway. Met3 have a dam that's the next one down. So feeding it under that dam will allow it to survive. Feeding it anything above the dam will cause it to pile up behind that dam and not resolve the issue. So this is how the analysis works. So if you can relate to that, I'm sorry I took so long explaining it, but that's how it works in my mind. So this nicely segues into something in genetics that's very important. It's the principle of epistasis. Epistasis is basically a biochemical phenomenon where the effect of one gene is dependent on the alleles of other genes. Another way to look at this is where the effects of one gene are masked or hidden by other genes. A third way to look at this would be where a mutation in one gene in a pathway may prevent the production of the end product. One method in order to identify epistatic relationships is to look at gene ratios, phenotypic ratios from genetic crosses. There is a relationship between these phenotypic ratios and the type of epistasis that's taking place within that system. The patterns are a result of different product interactions in these particular pathways. Now, there are 6 ways that epistasis could affect the predicted ratio of a dihybrid cross. So remember from previous chapters, when you have double heterozygotes and you cross them together, then the expected ratio is 9 to 3 to 3 to 1. And any departure from that ratio is clear indication that you may have epistasis. Here are the essentials of that type of analysis. So the very first column tells us that the ratio is 9 to 3 to 3 to 1. Then we have 1, 2, 3, 4, 5, 6 alternative ratios that can materialize when you have epistatic interactions. We're going to look at each one of these over the next few slides. Consider this example with budgerigars. We're going to study 2 traits, 2 characteristics controlled by 2 genes, the B gene and the Y gene. The B gene produces a blue pigment for the feathers, and the Y gene, a separate gene, produces yellow pigment also for the feathers. So the interaction of both pigments will color the feathers a particular color. So let's look at that key. The key is that wild-type birds that produce normal blue and normal yellow pigment will have green feathers. Whereas if there's a defect in one or the other, then the feathers will be appropriately colored, either blue or yellow. And if both pathways, the yellow and the blue, are defective, then the feathers are going to be white. So that's the relationship between these 2 genes. If we take 2 heterozygous, double heterozygous birds and we breed them together, then in the next generation, we get the 9:3 to 3:1 ratio. That tells us that there's no interaction going on between the two genes. The two genes are acting independently on two different pathways without interference. You should examine the outcome, the phenotype of the progeny of these Punnett square, and be able to relate to everything in this diagram before you move forward. Make sense of every step. If one step doesn't make sense, then you need to go back to other chapters to re-understand this knowledge. This is very important. In another historical experiment, Bateson and Punnett, yes, that Punnett of the Punnett square, they crossed pure breeding strains of white flowered sweet peas. And they found that all the F1 were purple flowered. And they crossed the F1 together to yield the F2. Now in the F2 they were expecting a 9:3 to 3:1 ratio, but what they observed was a 9:7 ratio. 9 purple flowers for every 7 white flowers. By looking over their Punnett square, they were able to quickly identify that the two genes were interacting. Not the alleles, but the two genes responsible for the flower color. From this, they concluded that the two genes work in tandem to produce the single product that's necessary for coloring the flowers, whatever color they are. So the process they called complementary gene interaction. Once again, the biochemistry of this biochemical pathway helps us understand the eventual outcomes. So if both enzymes are working normally, then you have a biochemical product that leads to the purple color. If one of them is blocked, then there's no pigment. If the other one is blocked, there's no pigment, because the precursor has to go through each of these steps in order to become white. So going back to the analogy of the mountain with the reservoir and the ocean, so water leaving the reservoir gets through this step, but it gets blocked by this dam. So no water is materializing at the ocean. And in this genotype, the dam is blocked early on, but the effect is the same. Water piles up behind this, therefore there's no water in the ocean. And in this case, both dams are defective, therefore there's no way that any water will get through. So in all three cases here, there's no water in the ocean. With duplicate gene action, the ratio is 15:1. So only one of the boxes doesn't produce something, the others do. And this is because you have two genes that duplicate each other's biochemical processes. So if one is damaged, the other one can take over. So going back to our analogy, we have two rivers flowing from the same reservoir, and they both make their way to the same ocean. So looking at this from a biochemical perspective, here we have two products, two enzymes, feeding onto the same step in this pathway. Just the double homozygous recessive genotype, where there's a block in both enzymes, is enough not to allow either river to feed the ocean. Therefore, that's the one box out of the 16 that produces white. The others are duplicate. The next category is dominant gene interaction. That produces a combined 6 in the middle, and you still get the 9:1 ratio on the extremes. So dominant interaction takes place because of the way that genes communicate with each other through their biochemical products. And this is illustrated by summer squash. So summer squash can exist in three shapes of seed. You can have a disc, you can have a sphere, or you can have a long seed. So depending on the combination of alleles of two genes, that's going to change the shape of the fruit. Let's look at the data. If you take a double heterozygous parent who has for each gene one dominant allele, they will have the phenotype of a disc-shaped seed. So you breed these together, and then you look at the offspring, and you'll find the following scenario. If either pathway is producing something, you will never get a long-shaped seed. If both pathways are blocked, you will always get a long shaped seed. So this one's easy. 2 blockages in 2 separate pathways which need both these products to perform a spherical outcome. So this is the default pathway. If one of the genes has a dominant allele and is functional, then it will produce at least one protein that will make it spherical. It doesn't matter whether it's protein A or protein B coming from either gene, there's at least one dominant, and that's enough. If you have both dominants, then you'll get the added effect of producing a disc. So let me go back to this. You need not at least one dominant, you need one dominant and not the other. Here you need one dominant and not the other. So that's the difference between the two. So this will give you the 9 discs. This will give you the combined 6, 3 from this, 3 from this, 6 spheres, and this one box out of the 16 will give you the long. We also have recessive epistasis. And in recessive epistasis, the last 2 ratios, the 3:1, combine into a 4 ratio. If one gene is present in the homozygous recessive condition, then that will mask whatever phenotype exists at the other gene, therefore giving you that ratio. So let's have a look at that. I think the example itself, when you look at the biochemistry, really will make sense. So the classic example is looking at Labrador retrievers, these are dogs, and their production of coat color in their hair filaments. So eumelanin production is what we're trying to follow. But not only that, the production takes place under the skin, but the pigment then has to be carried to be deposited in the hairs. So there's a factory and then there's a transport system. So this is a great analogy. So the factory can produce products, but it doesn't matter if they're not delivered to the destination. So keep that in mind as we go through this. So dogs that cannot deposit the product, eumelanin, that's produced by pigment cells, have a yellow coat color, regardless of whether the factory is working or not. Let's assume that the E gene is the truck and the B gene is the factory. So once again, if the trucks are not working, then there's no delivery of product. So when you have this condition, look at the E's. They're homozygous recessive. So that means the transport company is not functioning. In this example here, it doesn't matter what the B is, whether the factory is working or not. The trucks once again are not functioning. So in both these cases, there's no delivery of cargo from the pigment cells to the hair shafts. So the dogs are going to be yellow. In this case here, there is at least one truck working in each case. So here there is delivery of something to the surface hair shafts, and in this case it happens to be something that is produced here, and that's going to be a brown type of pigment. And in the case of the factory and the transport truck system working, then the factory produces a black pigment, and that's delivered to the hair shafts. So the thing here is that in this case the B gene can't produce black, so it produces a precursor called brown. The next ratio is due to dominant epistasis. So here we have the 9 combining with the 3 to give us 12. So in dominant epistasis, a dominant allele at one locus will mask the phenotypic expression of the other alleles at the second locus. So once again, going back to the summer squash, this time looking at a slightly different system, we can see how that works. The first gene affects the production of a green intermediate from a colorless precursor. The most important thing to realize here is that the dominant allele of that gene prevents the conversion to the green intermediate. So it blocks it, maybe diverts it to something else. And the second gene takes the green intermediate and converts it into yellow. We have a sort of inverted biochemical pathway. We have 2 double heterozygous parents crossed together with each other, then we would have expected a 9:3, 3:1 ratio. But what we're getting is a 12:3:1 ratio. So we have to explain what's happening here. Keep in mind what we said, that the dominant allele prevents the production of pigment. We're not used to seeing this. In our minds, a dominant allele will make an enzyme that converts this to something. But in this case, the system works in the opposite direction. So every time you have a W, a big W, that's going to prevent the production of pigment. So it doesn't matter if the enzyme for converting that pigment to yellow exists. If there's no pigment to convert, then there's no yellow color, and the seeds will be white. Once the lowercase allele is present in the homozygous condition, only then will you get green pigment. And thereafter, then it depends on the second gene. So the first gene, its condition, is masking the genotype of the second gene. Here it's not allowing it to do its job, and here it will then decide which of the two alleles of the second gene influence the offspring. And the last class of epistasis is dominant suppression, where the ratio is 13:1. In this case, the answer is straightforward. The dominant allele at one locus completely suppresses the phenotypic expression of the alleles at the second locus. In the example provided, we're looking at the flower color of a plant. So in order to produce blue flowers, you must have the L allele present. If you have the homozygous recessive condition, then the flowers will produce no pigment and are going to be white. So in this case, the dominant allele of the second gene, D, can suppress the color producing effects of the L gene. And this leads to white flowers in both L and LL. We're looking for genetic genotypes that contain the big D allele. One copy. That's all it takes to block the production of pigment delivery. So a big D in any one of the offspring will lead to a white phenotype. In addition, if you have two small L's, then the pigment will not be converted to blue. So the only thing that works is if you have two small D's and then you have at least one big L. Then you'll get the production of blue pigment. And that only takes place in these type of progeny. I like to use the analogy that we had earlier, but revised slightly with respect to the factory and the delivery trucks. So in our first case earlier on, we had the factory producing the pigment, and then the trucks were delivering it to their destinations. In this case, we have slightly different scenario. We have trucks bringing raw material to the factory, and the trucks are the allele big D and small d, and the factory is the gene L. And the factory only works if you have a big L, otherwise the factory is not working. And the trucks only deliver if there is a big D, otherwise if there's a small d, the trucks are broken. So you can see, you can explain this with that analogy. If you're not bringing in raw material to the factory, it doesn't matter if the factory is working normally, it can't produce product. Putting these two concepts together, then you'll have the answer to this problem. The last section of this chapter looks at complementation analysis. And this is a form of genetic analysis that allows one to determine if two mutations are in the same gene or in different genes. A deeper application of this principle then allows one to determine how many genes are responsible for the mutation observed, if the answer is other than one. Let's consider two terms first before we look at the analysis. The first term is genetic heterogeneity. This is when mutations in two different genes can produce the same or very similar mutant phenotype. And the second one is genetic complementation analysis. That's performed by mating two pure breeding mutants for these two similar phenotypes and then observing the outcomes in the first generation, the phenotypes of the F1. If complementation occurs, then wild-type offspring are obtained, and the mutations are said to be in two different genes. When the mutations fail to complement, the offspring have the mutant phenotype still, and the mutants are said to be affecting the same gene. In this analysis, suppose we have 4 different strains of mutant flowers. They each produce white flower types. And the question is, are the 4 mutations in the same gene or different genes, and how many genes are there? And the way that you solve this problem is you take pure breeding varieties of one mutant and you breed them together with the other. So these are pure lines, and you breed them together and you see what happens. And in this case, we produce purple flowers. When you take A and breed it with C, you get white flowers. If you take B and you breed it with D, you get white flowers. So in this case, the original mutation, the white, has not been prevented. So there's no complementation. Same thing here. But in this scenario here, the 2 mutants, when bred together, they restore the wild type condition, which is purple. So this must indicate that the mutations are in different genes, both of which lead to the white phenotype. But when you breed them together, you get double heterozygotes. Therefore, each gene now produces the necessary chemistry to lead to the production of the purple outcome. We can further deduce from this experiment that if A and B are in different genes, then what else belongs to the A gene? Well, when you cross the A gene with the C gene, it doesn't solve the problem. Therefore, A and C must be mutations in the same gene. Likewise, when B is crossed with D, it doesn't solve the problem. Therefore, B and D must be mutations in another gene. So A and C belong to one gene, B and D belong to another. Any combination between them will solve the problem. Thus, in this manner, you could have lots of mutations, all giving rise to the same phenotype. And by crossing the different mutations together, once you have pure lines, you can then answer the question, am I getting complementation or am I not? Now, if you're not getting complementation, then the chances are that the mutants must be all part of the same gene, therefore the same complementation group. So complementation, no complementation. No complementation leads to the finding of all the mutations in that gene from those available. So applying this to the Drosophila eye color mutants, we have different colors of eye color in Drosophila, and the question is, how many different genes do we have? Because some of those mutations may be affecting the same exact gene. And once you do the analysis here and you look at the data, you will find that we end up with 5 complementation groups, 5. And these colors here all belong to the same gene. So different places in the same gene being mutated will have produced the same complementation group.
The complementation test, also called the cis-trans test, is used to determine how many genes contribute to a particular phenotype. Cis denotes mutations in the same gene or complementation group. Trans refers to mutations in different genes or separate complementation groups. Consider a simple biochemical pathway in which a precursor is converted to an end product B in 2 steps, with each step catalyzed by an enzyme which, in turn, is specified by a gene. Gene A specifies the enzyme to convert the precursor to intermediate A, and gene B specifies the enzyme to convert intermediate A to the end product B. A mutation in either of the genes will prevent the end product B from being synthesized, giving the same phenotype. If B is an amino acid, for example, the mutant strains would be oxotrophic for substance B, and if this was a microorganism, B would have to be added to the growth medium in order for the cells to grow. To turn this around, if we isolate a set of mutants that require substance B for growth, we can find out how many genes are involved by using the complementation test. We will see how this is done using yeast as the model organism. The complementation test is done by mating two haploid yeast cells that are auxotrophic for substance B and determining whether the resulting diploid is auxotrophic or if it can grow in the absence of B. If one mutation is in gene A and the other mutation is in gene B, then in the diploid there is still a normal copy of gene A and a normal copy of gene B. So, normal enzymes A and B are both made. We say that complementation of the two mutations has occurred. Each has made up for the other's enzyme deficiency so that, between the two, a complete set of enzymes for the pathway is made and end product B is produced. This result means that the two mutations were in different genes involved in the pathway. If both mutations are in one of the genes, here A, then in the diploid there are still two normal copies of gene B, but there is no functional copy of gene A. So, normal enzyme B is made, but no functional enzyme A is made. In this case, no complementation occurs. That is, since both mutations are in the same gene for one of the enzymes in the pathway, there is no way for the pathway to be completed, and end product B cannot be made, and must be provided in order for the cells to grow. In practice, the analysis is done in the opposite way. If complementation occurs, then the mutations being tested must be in different genes that control the phenotype. If no complementation occurs, then the mutations being tested must be in the same gene.
Thank you. That concludes the chapter on gene interaction.